Hello everybody, this is Physics 1020-1090 lecture for Monday, March 16th. We're in chapter 24 on wave optics. This is the fourth video and last video for Monday, March 16th. It's going to be about diffraction gratings. First, let's review a little bit from Young's experiment. So that would be, forget this part, and let's look just at this diffraction grating picture. This is you have a double slit, classic Young experiment. And what you see then from the double slit are a pattern of interference fringes where you get constructive, constructive, constructive interference with destructive in between. That's just exactly what we saw on Wednesday of last week for Young's experiment. Suppose instead of 2 slits, you now had 3 slits. So there's 1, 2, 3 slits. What do you get when you shine light through there onto the screen? Well, you get another pattern of interference fringes. And these fringes are located at the same spot as they were for 2 slits. However, they're somewhat narrower, and in between each bright spot is a little small peak in between them. So you've gotten the main peak's gotten narrower, and the in-between spot has gotten a small little peak. Well, that's interesting. Wonder what happens with 3 spots. Well, let's go up to more spots. Let's go up to 5 spots. 5 spots is what we have here. We have 5 holes in the screen, and what you get is the same interference fringes, but they're narrower still and with tiny little peaks in between them. Well, take this to its logical conclusion. We're going to go up to having, by the way, these when I'm working with this camera, my left and right somehow get mixed up because the camera versus things, and it's really hard for me to point at stuff, especially when I'm looking from the back. But here now is a whole bunch of slits, maybe 1,000 slits, and they're going to be all the same distance apart. And what do you get? Well, you get a series of bright spots with the same separation as you did for 2 slit interference. But they're extremely narrow, and there's a whole bunch of tiny little maxima in between them, but they're so small that they can't be seen. So what you get now is it's just like taking the Young's double slit experiment, and it has now writing it in terms of very, very narrow bright peaks. And that's what that is called, is a diffraction grating. And I have a diffraction grating here, which you can see as I put up here. You're going to see various peaks inside me. You can see the green dot that's next to my web camera, and you can see various peaks next to it. So I turn it around, I get them in the opposite direction. Diffraction gratings are interesting. In particular, they're interesting because if you remember, the position of these peaks depends on exactly the color of light. So a diffraction grating will act like a prism. If you shine white light through it, you'll find different colors having peaks at different positions, but they're very widely spaced and they're very sharp. Diffraction gratings are used a lot in spectroscopy, where an atom gives off a certain number of wavelengths, and you can split them up and do very careful measurements using a diffraction grating. Let's derive the equation for the diffraction grating, and this is going to look very much like Young's double slit analysis. Here we're going to have a whole bunch of slits, and in order to get this very large constructive interference, the path length of the light between one and another of the rays has to be just 1 wavelength. If it's 1 wavelength difference, or 2 or 3, some integer number of wavelengths, and it's going to be that many wavelengths there, it'll be that many wavelengths another one, each one will add up They'll all be in phase because each one is an integer number of wavelengths extra path distance. You'll get constructive interference. You'll not get constructive interference of 2 rays. You'll get constructive interference of thousands of rays. So you'll get the diffraction pattern. We can make this picture by making a small triangle here. This side is D. We're going to trying to find what this extra distance is. We'll drop a perpendicular line, and we're trying to find the extra distance of that ray is delta R. And this angle in this triangle is theta, is the same as this angle theta. So what you have now is you have an analysis just like Young's experiment. Your delta R is d sine theta. That's just trigonometry. And you want for constructive interference to that be an integer number of wavelengths, m lambda. Lambda is the wavelength. M is 0, plus or minus 1, plus or minus 2, some integer. So when you solve this for sine theta, you get the angle where you get these bright peaks is going to be some integer m. That's going to be the order of the grating spots, the spots, times lambda divided by d. So you get what we saw for Young's experiment back, but it has a somewhat different meaning because of the narrow peaks. When is this used? Well, I told you it's used for spectroscopy, but there's another case where we see this sort of effect, and that is X-ray diffraction. Well, X-ray diffraction is using a crystal as its diffraction grating. So it's letting light go through a periodic array of openings, but in this case, those openings is a space between individual atoms, and that means the distance between slits is not half a millimeter or a micron. It's down to the size of atoms. Atoms are the size of an angstrom, or in other words, a tenth of a nanometer. So your atomic spacing is tenth of a nanometer. Turns out that if you want to see these effects, you really want to use a wavelength of light that is similar to the spacing of the slits, or the spacing of the atoms. If you want a wavelength of light that's tenth of a nanometer, you've got to go down into the X-ray part of the spectrum. So you shine X-rays at a crystal, and what you see then is going to be a whole bunch of very narrow bright spots, and the location of those spots is going to tell you something about the structure of that crystal. Now, let me show you, we're jumping ahead a little bit, but I want to jump ahead to chapter 27 in your book and show you a couple pictures. First of all, here's a picture of an X-ray diffraction pattern from a protein. This you can see it right here, or you can open your book and look at it. It is figure 27.7. What you see is you shine X-rays and you got a whole series of bright spots, and those bright spots correspond to where there's constructive interference. And then you can use mathematical tricks to figure out the structure of the protein from that X-ray diffraction pattern. So that's become a very important method in biology to find the diffraction, the structure of large molecules. The most historical example is on a couple pages later in your book. It's in figure 27.14. You'll find this picture. Not quite as nice an X-ray diffraction pattern, but very important historically. That's the X-ray diffraction pattern from DNA, first measured by Rosalind Franklin. That is the exact picture that Watson and Crick saw when they visited Franklin, and that gave them the clues they needed to figure out the structure of DNA. So DNA structure was discovered using this X-ray diffraction. So diffraction is a very important issue in not only light, but also finding structures of biological molecules. All right, so we have now finished the lectures for Monday, March 16th. We will next move on Wednesday, March 18th, and finish chapter 24 on wave optics. In particular, we'll talk about interferometers, and we will talk about polarization. See you then.